thank you all very much for joining us this morning and this afternoon in India. And this is the first of our two joint sessions between the National Biofilms Innovation Centre and the India Biofilm Society. And this morning we have 6 presenters, 3 from our Indian colleagues and 3 from the UK. And I would ask that if you have any questions or comments you wish to leave or make, then please put those in the Q&A box. As you've arrived in the meeting, you will be on mute, but we will see your questions, and those questions will be logged with the email you used to log in on. So if we don't deal with them today, they will all get dealt with and answered by the participants and the presenters post the meeting. So please do leave any comments, questions, or feedback in that Q&A box. As you know, today's meeting is joint between our 2 societies, and the purpose is for the 2 research communities to share their areas of interest and to highlight opportunities for collaboration, either between each other or those of you who are listening in and watching in. Today, we have a packed agenda, as they say, and we're going to start off with our 3 speakers from the India Biofilm Society and then 3 from the UK. I will, to begin, hand over to my colleague Snehal Kedan, who's from the India Biofilm Society and the University of Pune, to introduce the first 3 speakers.
All right. Hi, everybody. My name is Snehal, and I will be introducing the speakers from the India Biofilm Society. Our first speaker is Dr. Karishma Kaushik from the Savitribai Phule Pune University in India. And she's going to talk to us about the human relevant approaches for biofilm infection states that her lab has been taking. So Karishma, you can go ahead and share your screen.
Okay, thank you. Just give me a second. Shall I get going? All right, so good morning, good afternoon. Thanks for the opportunity. I'm Karishma. I'm an early career independent investigator at University of Pune. I returned to India in 2018 on a re-entry fellowship from the Government of India that supports my 5-year research agenda. This is a broad overview of the work we do. We are one of India's very few laboratories working to develop human relevant approaches in the infection space. And our particular focus is biofilms, and that's what brings us here. So this you can look at later, but I'm going to be giving you a snippet of a few stories over the course of this very tight 10 minutes that Mark and Snehal have allotted us. All right, so talking of biofilms in detail here is a bit like taking coal to Newcastle, you know. So I'm going to skip this and say that we work on wounds, and the most common cause of non-healing wounds is biofilms, often with the pathogens Pseudomonas and Staph aureus. So this is how we look at the wound infection microenvironment. We are trying to bridge this in vitro, in vivo divide, and we look at it as a bioengineering problem. We think of it as a two compartment system with the host, the immune system that interfaces, and of course there are a host of factors that contribute to a very unique milieu. So how can we incorporate these factors in vitro to make our systems more human relevant? So current model systems in vivo and ex vivo are challenging and cumbersome, and the in vitro, well, they're highly reductionist. So what are some of the approaches we adopt? One of our approaches is to develop a biomimetic wound fluid milieu. So we have this proprietary formulation that we are working to publish soon, and we are studying interspecies interactions and biofilm formation and susceptibility in this milieu, which is an advancement to standard fetal bovine serum that is used to mimic the host state. So with very early data, you can see that it differentially affects the 2 pathogens, Pseudomonas aeruginosa and Staph aureus, as well as it shows differences when compared to FBS for biofilm formation as well as metabolic activity, so the living component of the biofilm. When we try and look at the 3D architecture, so this is not a tiles, this is a tile scan, this is the whole biofilm that we look at. We see that biofilms formed in our proprietary milieu are much thinner than FBS, and certainly compared to standard laboratory media. So we think this is important because it may be when we are testing antibiotics or antimicrobials in these systems, FBS and LB, we are actually overestimating the biofilm. and could have implications on our susceptibility results. Another approach we are taking is to co-culture the wound bed under human relevant conditions so we can reduce the dependence on mice and rodent models of wound infection. So the two important cells are the fibroblasts and keratinocytes, and there's a discrepancy in co-culturing them because one of them needs serum and the other, as luck would have it, prefers to be serum-free. So using your standard commercially available media components, we've co-cultured them, and you can see they're beautifully growing well together with their characteristic morphology, and reduced the serum to 1 to 2%. Not only this, we don't have to add individual growth factors and supplements. We don't have to have the feeder system. So it's a really easy tool to adopt in the lab, and reducing the serum is important for couple of reasons, but before that, I'll tell you that the platform can actually be used to study cell migration and wound healing under these conditions, and both cell types appear to do very well. Reducing the serum is important to be able to co-culture microbes, which we want to do very soon. So where do we see us taking all of this in this research agenda of our group? We want to look at developing an in vitro platform that can incorporate human relevant in vivo components and yet not be in an in vivo model. So we have this handheld biofilm on chip or wound infection on chip device, which will have the co-cultured wound bed, which we've just published with microbial elements, and we flow our proprietary wound milieu through it. So we're not flowing just cell culture media. We can use this under shear conditions, which is what we see in clinical states, to better understand host microbe interactions, as well as to rapidly screen and test therapeutics. So we don't think we can replace animal testing with this platform, but we definitely think we can have this preclinical step, which is like a go, no-go decision before you take it onto animal models, given we know that their translation is definitely not as optimum as we would like it to be. So we are a young, but very, very dynamic group. We have 3 independent grants, 2 from the Department of Biotechnology, and we look forward to engaging and growing and taking biofilms in India forward. Thank you.
Okay, if anybody has any questions, you can type them into the Q&A box, and we might have time to answer 1 or 2.
Thank you, Snehal. Was I on time? I take that. Yes. I was on time. Okay. I take
yes,
that means I was on time.
Yeah. All right. If any of the panelists also have questions, I think you guys can just unmute yourself.
Yes, Helen says a microbiology wound community. Thank you, Helen. And Snehal is nodding her head. Yes, that is our next step with the wound bed that we've co-cultured the 2 cell types. You want to add microbes to the system. It's going to be like a controlled contamination. But yes, that is what we are trying to do.
If I may ask a question, I think very interesting presentation by you, Karishma.
Thank you,
this is Saurav. You will probably hear me later today. Preparing this wound bed is, I think, quite challenging, and I'm nowhere near that. I'm an engineer. So that's probably something that we might want to talk more with you. So once you hear my presentation, maybe you can comment more on how you might be able to help.
Sure, sounds good. Yeah. All right. What is the throughput of this in vitro system? Well, let me be honest, the platform and the peristaltic pump have to be assembled. They have to be in a CO2 incubator. You have to take it to the confocal. So I won't really say it's rapidly high throughput, but I definitely think it's more throughput than animal systems, certainly, in terms of the selective, the control you have over it and the reproducibility.
Karishma, may I ask a question?
Yes, sure.
Will this wound bed interfere with confocal microscopy when you are inoculating microorganisms in it also?
No, we don't think so. If they're differently tagged, we should be able to see them all. The wound bed can be immunostained and the microbes are fluorescent. So no, I don't think so.
Okay, right.
Yes.
Okay, I think we can move to the next.
So yeah, there's one more question. Any other questions, please keep typing them in and they will get answered subsequently. Thank you.
Okay, so there is one question in the chat window for you, Karishma. I think you can type in the answer then. Okay, So our next speaker is Dr. Deepti Jain, who is from the Regional Center of Biotechnology in Faridabad, and she's going to be talking to us about transcriptional regulation of biofilm-related genes at her lab studies in Pseudomonas.
Hello, everyone. So I'm Deepti. I'm an associate professor at Regional Center for Biotechnology, which is in Delhi NCR area. And before I begin, actually, I would like to thank Dr. Karishma and Snehal for bringing together the Indian biofilm community. And I would also like to thank Professor Mark and NBIC for giving us this opportunity to share our research work with you all. So we are a structural biology lab, and the research in our lab focuses on structural studies of macromolecular complexes that are involved in regulating gene expression, particularly in pathogenic bacteria. And we extensively use structural biology tools such as X-ray crystallography, small angle X-ray scattering. We use a lot of biophysical tools like dynamic light scattering, biochemical assays, in vivo assays, complementation in bacterial systems, followed by transmission electron microscopy and looking at gene expression through qPCR. We also do a lot of in silico inhibitor search using computational biology tools, molecular modeling, docking, and MD simulations. So lab interests currently are on 3 different bacteria, which is Bacillus subtilis, where we have determined structures of complexes between the transcription factors and DNA. We have also looked at some of the proteins involved in antibiotic resistance in Staphylococcus aureus, but the primary focus in the lab currently is on understanding regulation of biofilm and flagellar gene expression in Pseudomonas aeruginosa, where we have determined structures of some of the activators, anti-activators, and more recently their complexes. Now, as you can imagine, that we require synchrotron access for us to be able to collect data on these crystals. And currently India does not have a synchrotron facility, but we do all our data collection at the ESRF, European Synchrotron Radiation Facility, which is present in Grenoble, and the access is provided to us through Department of Biotechnology, Government of India. We also have access to Electra Synchrotron in 3S, where we are using XRD2 beamline, again sponsored by Department of Science and Technology, Government of India. So this facility is open to all the Indian macromolecular crystallography groups. And before the pandemic, we were not only sending our crystal samples, but we were sending young researchers, students to get trained into using these facilities. The model system that we use in our lab is Pseudomonas aeruginosa, which is gram-negative monotrichous. And as you all would be familiar, that this is a big cause of mortality in patients who have immunocompromised systems, such as those suffering from cystic fibrosis, burns, wounds. And the persistence of Pseudomonas is because of its ability to form biofilms, and no drugs currently are available. So the regulation of biofilm formation in Pseudomonas is a complex process that it involves a lot of pathways, and including quorum sensing, 2-component regulatory systems, and which are intertwined and result in the expression of exopolysaccharide production, which is the matrix of the biofilm. So these, we want to look at the atomic details of these molecules. What are the structural transitions that are happening when these transcription factors get activated or phosphorylated, and when they interact with the DNA, and so on and so forth. So amongst this pathway, we are currently focused on the genes which are responsible for production of these exopolysaccharides. And in case of Pseudomonas aeruginosa, it is largely the PSL, PEL, and alginate. So the genes, you can see these are the exopolysaccharides of the Pseudomonas biofilms, and the genes which are responsible, there are about 30 odd genes which need to be overexpressed for the biofilms to form. So the transcription factors which are regulating these genes, in our lab, we've seen that if you mutate these transcription factors, some of these are co-regulating motility and biofilm formation. So one such transcription factor that we studied in the lab, when we mutate the single amino acid based on our structural studies, when we mutate single amino acid, this can render the organism aflagellate. And when it's aflagellate, we see that it has drastically reduced ability to form biofilms. So we implicate the role of flagella in biofilm formation because it acts as a mechanosensory organs. It promotes surface adhesion. It is a component of the matrix. And we also have a multiflagellate phenotype bacteria, which also shows somewhat reduced ability to form biofilm. So these are based on the structural studies, and we also have done motility studies on these mutants. As you can see, the mutant which is defective in biofilm formation or in flagella formation is also defective in motility. So this is the one which is, we are measuring the diameter of these zones and corresponding that to the ability of the bacteria to move. So one of the regulators that we have looked at in our lab is called as FLEQ, which regulates both, like I said, co-regulates flagellar gene expression and biofilm gene expression. And if it's knocked out, all the biofilm, the flagellar genes get downregulated. And it also acts as a repressor of biofilm. So if you knock out, the biofilm related genes are upregulated. However, in the presence of a second messenger like cyclic di-GMP, which also binds to FLEQ, this protein undergoes conformational change and then starts to activate transcription of biofilm genes. This is an ATPase, so we measure the ATPase activity in our lab, and when you knock out the ATPase activity in one of the mutants, they have reduced ability to form flagella. So this is qPCR showing flagellar genes get downregulated when ATPase activity is knocked out. We have solved the structure of this protein in complex with ATP, and so ATPase activity, if it is inhibited, there is reduced ability to form flagella. There is reduced ability to form biofilms. In literature, we also have structure of this protein in complex with cyclic di-GMP, which I said was a secondary messenger. And this is an overlay of the two structures, so the ATP bound structure and the cyclic di-GMP bound structure. And we can target both these sites for the search for anti-biofilm compounds. So we're doing that in silico, and through in silico, we have shortlisted a few compounds which show at least more than 50% inhibition and ability to form biofilms. We are characterizing these further. We have done these assays in vitro. We have seen that these compounds also inhibit the ability of FLEQ to hydrolyze ATP, and we are characterizing them structurally. So we will make co-complexes of these compounds with the target proteins. We are seeking collaborations in the field if anybody has animal model systems to try out the mutants that we have created in the lab, or somebody with chemical expertise on chemical synthesis where we can derivatize these compounds further, or in clinical microbiology, or almost anybody who's interested in looking at protein structures. So this is my group, and I take this opportunity to also thank my amazing students, postdocs, junior research fellows. I want to acknowledge funding from all the Indian government organization. And once again, thanks India Biofilm Society for this opportunity. Thank you. I'll be happy to take questions.
Okay, thank you so much, Deepthi. If anybody has questions, you can go ahead and type them into the chat window. I think we have time for maybe 1 or 2 questions.
Okay.
Okay, I think if there are questions, you can either answer them in the chat window, Deepthi, or we'll forward them to you.
Sure. Sounds good. Thank you.
Okay, our next speaker is Dr. Ankita Singh, who is from the A.D. Patel Institute of Technology in India. And she's going to be talking to us about the work her lab has been doing to use DNA to prevent and eradicate biofilms.
Good morning and good afternoon to everyone. And I'm so happy that we are starting this everything related to biofilm on the auspicious day of Teachers' Day celebration. So happy Teachers' Day to everyone.
Thank you.
I shall be starting my screen. And Snehal, please ping me whenever there are like 8 minutes over.
Sure, okay, I will do that.
I'm basically working in the Department of Food Processing Technology with PhD in dairy microbiology. So I'm like versatile with foods and pathogens and enteric infections also. My PhD work was also in biofilms. So this time I'm working with the enzyme DNase and its efficacy to prevent and eradicate biofilms. So I'm so glad to say that I don't have to introduce biofilm in this collaboration. So let's skip the introduction to biofilm. And this is the structure so far. In this concept, I'm targeting the extracellular DNA, which acts as a cementing material for the biofilm structure. It has several functions enlisted in the literature so far published. So working with DNAs, we are targeting both things, pre-treatment, that is prevention, and a post-treatment, that is the eradication of previously formed biofilm. For this, we want to mention that degrading the biofilm matrix will help in penetration of the antibiotics or biocides and then combating the biofilm related microbial drug resistance and the infection virulence. So for validating the hypothesis, we added calf thymus DNA, then extracellular DNA, which was extracted from the respective organism biofilm, and then genomic DNA of the respective organism. So far, we observed almost 180% increase in biofilm formed by the pathogen. This was again dependent on species to species. So coming to pre-treatment, that is the prevention of biofilm. DNase was added in the broth itself. And we optimize the concentration on the basis of biofilms allowed to grow up to 96 hours. So 10 microgram per ml was optimized as the concentration. With this, we did confocal microscopy and we observed that there was a reduced surface coverage and a reduced height also. So overall, our prediction is that a biofilm matrix might have destabilized, but we did not have any conclusive images from scanning electron microscopy. That would have been more helpful to interpret that the matrix itself was damaged. Now, eradication of pre-formed biofilm will formed in control, and then with the various time limits, DNase was added. So DNase was considered as again 10 microgram per ml, and we observed that irrespective of the age of biofilm, there was 70 to 80% reduction. So that was like one optimistic point that post-treatment seems to be better with Pseudomonas aeruginosa PO1 biofilm. The contact time could be reduced with only 5 minutes and still 80 to 93% reduction in biofilm, especially in presence of magnesium. But magnesium is expected to interfere with the antibiotics. So when DNase is incorporated along with antibiotics to combat the biofilm, this may not helpful. But alone DNase should be helpful. So again, the confocal images, it indicates that the height was reduced, but this time the surface coverage was not reduced. But in comparison to the pre-treatment, post-treatment results in lesser height of the biofilm, that is almost 20 micrometer. We tried this with a consortium of pathogens formed in different proportion, and using a solo pathogen, we tried pre-treatment, post-treatment, and the combination of both treatments. the results were inconclusive. But when consortium was formed, it definitely showed that when Pseudomonas was double the other organisms, or when Salmonella was, or when E. faecalis, Staphylococcus aureus, or Klebsiella, in all treatments, post-treatment seems to be better with DNAs. So far, DNAs works with both as prevention and post-treatment. This may not be very helpful in case of food industry because a large quantity of DNAs may affect the cost of the system. But again, food safety is very necessary. So this may be incorporated in like ultimate regime in combination with other conventional methods also. Other findings of our projects are anti-biofilm effect of DNAs in combination with the antibiotics and biocides. Ongoing research includes anti-biofilm effect of foods and then biofilm-based microbial fuel cell. I'm also working on encapsulated biofilms of probiotics for its incorporation in extruded foods, which includes temperature of almost 140 degrees. So future scope for this, I'm taking up coating of materials, which may be catheters, prosthetics, or packaging material for DNA's application. And this was done under the funding by DBT. I need particular suggestion for the destabilized biofilm matrix because my results were not conclusive. I'm not sure whether WGA with CLSM may be helpful, and how to stabilize enzymes on the surface. That is again my question. My suggestion for this meeting, there may be a portal for sharing the information regarding the vacancies for students, especially because it would be better that biofilm trained students are in future also working in biofilm. Only then something will be conclusive for the whole world. Then sharing of open sources for softwares and collaborations, obviously. At national level, there are several hitches which I have personally observed. I feel that students, when they are going for analysis, either the rate or the way of reaching or the accommodation at the place where analysis needs to be conducted is a big concern for the students. So again, these things should be shared at the personal levels. So that's it.
Okay, thank you so much, Dr. Ankita. I don't see any questions in the chat window yet.
Dr. Ankita, could I ask a question? Thank you.
Yes, please.
Thank you for your great suggestions about enhancing collaboration, which we will certainly look into and build on. And I also had a question about DNAs. What do we know about its overall stability in terms of temperature and ability to be formulated? How robust?
It has been reported to be stable up to 80 degrees, but optimally it should be considered only 60 to 70 degrees. 70 degrees should be considered at max. And with pH it is stable only up to 9 pH. Beyond which, with biocides and detergents, it might not work well. So that is why it should be used as after the application of biocide.
Thank you. There is one question.
Yes, there is one question in the chat window. It says, did I understand correctly that DNase was only effective against multi-species consortia when Pseudomonas aeruginosa was present? And any thoughts on why this could be the case?
No, a consortium was developed with multiple pathogens in various concentration. If you can see this graph again, when we say that Pseudomonas aeruginosa 2x means that in the consortium, Pseudomonas aeruginosa was double the concentration of the other bacteria. In Salmonella 2x, it means Salmonella was double the other pathogens. Similarly, other cases also. In all these cases, when there was mixed biofilm, post-treatment worked out better. That eradication was easier.
Okay, thank you.
Okay.
There is one more question in the chat window, which I think you can type and answer. We will now move on to our next set of speakers from NBIC, which Mark will be introducing.
Thank you, Snehal. So it's my pleasure to introduce three speakers from the UK. And the first of these is Dr. Esther Karunakarn from the University of Sheffield, Department of Chemical and Biological Sciences. And she's going to be talking about SCARAB, in vitro and ex vivo models of biofilms. So Esther, over to you. Thank you very much. You're still on mute, Esther.
Yeah, I think I've now managed to unmute myself. So I'll share my screen now. Hello, everyone. Today, thank you, NBIC and the India Biofilm Society for this opportunity to present the research facility that I lead called SCARAB and some of the research highlights within SCARAB. So my name is Esther Karunakarn. As Mark said, I'm a lecturer in the Department of Chemical and Biological Engineering at the University of Sheffield, and I have been for the last 3 years, so I'm fairly new to academia. I'm also the co-director of an Innovate UK funded research facility called SCARAB, which I'll tell you a little bit more about later in the presentation. So in terms of training, I'm a microbiologist. I am from the south of India, a place called Tamil Nadu. I did a BSc in microbiology in India from Bharathiar University. And then I worked as a clinical microbiologist for a fair period of time. And then I sort of wanted to open up bacteria and look at their inner workings. So I moved to the UK to do a master's in molecular biology, which I did at the University of Leicester. And then I went on to do a PhD in biochemical engineering in the University of Sheffield. So I was quite fortunate, as Deepthi was saying earlier as well, that my PhD was on the topic of biofilms, similar to her, where she said her PhD topic was on biofilm formation. So so my work, my PhD topic was titled The Role of Autoinducer 2 on Bacillus cereus biofilm formation, which I completed in 2012. So I just, when I was preparing this presentation, I just looked back and I thought, yeah, that's 12 years of biofilm research, so which has sort of stood me in good stead in my lectureship, and biofilms are my topic of expertise. So my research philosophy is holism in biofilmology, in biofilm formation. So I published a paper outlining my research philosophy, which I titled Biofilmology, quite some time ago. So in that, I don't have to tell this audience, but I outlined the fact that biofilm formation is actually a multifactorial problem, and therefore we'll have to take a multidisciplinary view in order to tackle biofilms, to get a real understanding of how they form, how they maintain themselves, and how we can sort of manage their impact in everyday life. So we've got to take a multidisciplinary approach. So I've sort of taken the view that biofilm requires a trifecta of conditions in order to form, maintain, and create an impact. So this biofilm trifecta, I look at it as the surface to which the pathogen adheres, the cellular metabolism within the pathogen, and the environment that sort of the pathogen sort of feels influence is found within the environment. So within a particular environment. So these three sort of let's say, factors need to come together for a biofilm to form. So how can we study them? So I look at a plethora of different methods. So in order to study biofilms and the surfaces and the impact of surfaces in biofilms, I use a lot of techniques from soft matter physics and colloidal chemistry. So particularly techniques like zeta potential analysis, contact angle measurements, the XDLVO model, X-ray photon spectroscopy and FTIR, and to look at the intracellular processes and the extracellular polymer production. We use analytical chemistry methods, particularly mass spectrometry and mass spectrometry-based proteomics to study the intracellular environment and the extracellular polymeric matrix. So due to time constraints, I'm not going to go into a lot of details to do with sort of the outcomes of my research, but if anyone has any questions, please feel free to get in touch with me, and I'd be happy to go through all my papers with you. So when I received a lectureship, you know, I was thinking about holism in biofilm research and about practicing this, this holism philosophy. And it's a lot of fun to think about that. But then I realized that one of the major hitches in biofilm formation and a repeated reality is that the outcome of these experimental techniques is heavily dependent on how the experiment is set up. So for example, the culturing conditions really influence the outcome of experiments. So because biofilms, they're living and they do adapt. So when I was thinking about this, I was thinking if small variations in culture conditions can cause biofilms to change so much, and if biofilm formation gives us different results based on our culturing conditions, how is it realistically that we can translate what we see in the laboratory to manage sort of biofilms, or, you know, how can we translate what we see in the laboratory to actually what we see in everyday life? And how do we translate the management techniques that we develop in the lab to actually have a particular impact in the real world? So this is particularly evident when I was considering the problem of antimicrobial resistance, because, for example, if a drug can eliminate biofilms in the lab, that's great, but when we commercialize it and translate our findings to an infection scenario within the human body, does that actually work? And, you know, and in most cases, we're finding out more and more that it doesn't. And that's one of the reasons why antimicrobial resistance is increasing sort of in our society. We presented this sort of thinking to Innovate UK, and we put forward a solution. So Innovate UK is a funding body within the UK. It funds research, particularly translational research. And so we presented this problem to Innovate UK, and the solution that we sort of proffered was that we would create sort of the right culturing conditions for biofilms within the laboratory, so that whatever treatments that we develop, we can actually translate that more effectively to practice. So that was the solution that we offered, and they gave us close to $750,000 in British pounds in order to set up a research facility, which we call SCARAB, or the Sheffield Collaboratorium for Antimicrobial Resistance in Biofilms. So within SCARAB, the aim is to bring 4 different disciplines together, so microbiology, tissue engineering, immunology, and computational biology. So we bring these 4 disciplines together, and our aim is to create representative laboratory and computational models of biofilms. So we create these laboratory models and computational models of different infections within the human body. And once these models are created, what we intend to do with them is to use them as a platform for testing treatments and for developing new diagnostic procedures. The happy coincidence is that we can also use these models to study biofilm disease progression. So, unpick and uncover fundamental understanding of biofilms. So so I'm going to present a couple of examples of models that we have developed over the last few years. So the last 3 years that SCARAB has been in existence for. So the first model that I'd like to present to you is the ex vivo model of keratitis. So we've just recently published how we set these models up in the journal Jove. You can see the screenshot for that on the screen. And the picture of what the model looks like is what you see on the right-hand side at the top. So what we do is we source waste tissue material from abattoirs. So from the food chain, we have the waste material, so heads of pigs and eyes of pigs are at the moment in the UK a waste product and a byproduct of the food chain. So we source what is essentially a waste material and we process them in the lab to sterilize the tissue. We use that to set up biofilm infections. So you can see on the picture on the top right-hand corner, our control is a clear cornea, and then we infect that with different microorganisms. Pseudomonas aeruginosa is one of the organisms that we infect with, and after 24 hours, we find an infection. So this work was funded by the Medical Research Council, which is another research council in the UK, and we're using this model in order to develop novel treatments. And we've received funding from the National Biofilm Innovation Centre to work with companies to look at these different treatments. So this is one example of a model. So this model is used because it incorporates the 3D tissue structure, the biofilm, as well as a tear film in order to test treatments. So the next model that I'd like to present to you is a model of wound infections and catheter-associated urinary tract infections. We set this model up using this platform called the Bioflux, which is a high throughput microfluidic platform, which is interfaced with a fluorescence microscope. And then we can precisely control the culture conditions, including the flow conditions over the biofilm, the nutrient composition, as well as the gas composition that the biofilm grows. Can grow aerobic, anaerobic, and hypoxic biofilms. And we get lovely images and videos that look a little bit like this. So the picture that you see here is a model of the catheter-associated urinary tract infection. So this is a Proteus mirabilis biofilm. And what you can see here is that not only does the biofilm form, but we also get crystal development within the biofilm. So those are the crystals, are these structures that have like a black outline, but a clear middle. So these crystal formation in real time we can look at. The wound infection gives you videos like this, where we've coated the wound bed with skin keratinocytes. So the cells that you see here are skin keratinocytes, and then we grow them under very defined culture conditions, and then we infect them. And what you see now is that biofilms form, and you can look at them in real time. So we're using these to now look at the impact of treatments, and we're also using these to develop diagnostics. So to summarize, I'm Esther Karunakaran. I'm a microbiologist with a wealth of experience in biofilm research. I'm the co-director for this research facility. I develop in vitro and ex vivo models of biofilms. And I collaborate with clinicians in India in the LV Prasad Eye Institute and the Christian Medical College Vellore. And I hope to deliver breakthroughs in biofilm management through my research. And if you're interested in working with me, please do get in touch. Thank you.
Thank you very much, Esther. I mean, I love the idea of biofilmology. Thank you.
Thanks, Mark.
Thank you very much for a very interesting and insightful talk. We probably have a chance for one question if anyone had one they wanted to input or ask. But if not, and while you're thinking about that, please remember you can put them into the Q&A box, and all of those will be dealt with. And you can also email us at NBIC or India Biofilm Society after this meeting as well. So what I'm now going to do then is introduce Dr. Naveen Kumar Devanga Ragapathy, who's also from the University of Sheffield and is a Global Challenge Research Fellow. And he's going to talk to us about the missing link between antimicrobial resistance and biofilms.
So I think he's not
was there a moment ago.
promoting him to a panelist, because I don't think he was.
I think
Yeah.
Is he back?
Hi,
Yeah, thank
thank
you.
you. Thanks a lot for
Sorry,
the
I thought you
introduction.
were in and then you left and came back. Good, thank you.
Sure. So I'll share my screen now. Thank you.
Thank you.
So I hope everybody can see my screen now.
Indeed,
Yes.
we can. Thank you very much.
So, so today I'll be talking about the missing link between antimicrobial resistance and biofilm. So as Mark has introduced me, so I'm working in UK, actually in the Scarab facility in Dr. Esther's lab. So I have joined Scarab 1 year before. So by getting this Global Challenge Research Fellowship. So previous to that, I was working in the Christian Medical College, Velo. So where we happened to meet Esther and then collaborate and to take off this project. So, so I'll be giving a short introduction about like what I'm working on now. Okay. So the research problem that we have on clinical infections related to biofilms is actually in a considerable and significant amount. For example, if we see the biofilms that grow on the medical devices like ventilators and catheters, where like patients spend most of the time in the with the critically ill conditions in the highly dependent units. So most of those patients are ventilated. And it actually becomes a very practical problem for the clinicians to clear off the infections, because every time when you use a catheter or a ventilator for a prolonged period of time, so it tends to form the biofilm and then it tends to form a secondary infection. So that is how it's becoming very difficult. And actually, if we will be able to treat biofilms properly, it can prevent many otherwise preventable deaths. So as of now, with the statistics, so we know that the 65% of the hospital-acquired infections are due to biofilms, and approximately 80% of the medical devices in the patients are associated with biofilm infections. So in the UK, actually, the biofilm treatment costs about £1 billion for the NHS annually. So, and the research project which I am taking now try to address two UN Sustainable Development Goals of good health and well-being and reduce inequalities for the treatments. So the hypothesis that I had in mind is like, as rightly pointed by the previous speakers, Esther and Karishma. So there is always there is a lack between the ability of us to study exactly mimicking the in vivo conditions. For example, like in this problem, so we have a problem of antimicrobial resistance to clear off from the patients, and there is also an additional challenge of biofilm which is growing. So both go hand in hand, compatible to each other, and then makes the situation worse. So it is often like neglected due to the inability to mimic the in vivo situation. So biofilms are very less read. So especially when it comes to patient treatment care. So we were thinking of a laboratory tissue culture model. So to mimic such conditions, so this will act as a platform to evaluate the antimicrobials that we are currently using for the treatments, to know whether the antimicrobials that we give for treatment actually work on biofilms or not, and what is the impact of that on the output of the patient. So the research objectives are in two cases, like, so we are trying to attempt to identify the microbial diversity among the biofilms. So especially in patients with ventilator associated pneumonia and catheter central line associated bloodstream infections. So where it is related to the catheters. So, and to investigate the cross resistance in biofilm environment. So whenever we talk about biofilms, it's a complex microbial community, as you all know already. So there happens to be like the specific mechanism for exchange of antimicrobial resistant genes between the species. So it is not always a single species that is forming a biofilm. So there is always a chance that there are multiple species together causing a co-infection, which forms complex biofilm. So there happens to be the transition or transfer of antimicrobial resistant genes between each other to come across that stress situation. So we are actually taking up to do metagenomics to identify the population, like what is there in the population, to see the microbiome, virulome, and the resistome capacity, to see like what are the important factors that is responsible or that is facilitating this transfer. So, and also we are looking at regulation of this AMR and biofilm factors, so like which influence one over the other. So this, for this we do transcriptomics, like it's a meta-transcriptomics, and also the single species and polymicrobial biofilms. in vitro, mimicking the in vivo conditions. So we are actually working on a model to a lung airway model. So to test the single species and polymicrobial biofilms for their susceptibility against the current antimicrobials. So this will help us to identify specific markers, so which can be used for diagnostic purpose. For example, if you're going to quickly know whether you got an infection from a patient in a critically ill condition, you got a susceptibility test. So whether you want to go directly for the treatment and define the dose based on the MIC levels, or you want to see whether you're going to know whether it's a biofilm forming agent or not. Because if it is going to be a biofilm forming infecting agent, then it is very difficult to clear the thing. So you need to adjust the dose accordingly. So to enable that critical point, so we are trying to develop a quick PCR panel to know whether the agent, means the infecting agent, the pathogen, is biofilm forming or not. And also we will evaluate the current and emerging alternatives for the biofilms. I'm sorry. So this is the thing which I'm working on currently. So developing a air-liquid interface for human lung model. So what we do here is we take off the lung cancerous cells. So because cancerous cells are we can work with it for a prolonged time. So we have, and it is also stable. So we have selected the lung cancerous cells, and then these cancerous cells are grown in transwells. So initially we allow them to grow in a submerged condition. So after a few days, so then we supply it with a special medium called pneumocult ALI medium. So where it has got growth factors. So it actually enables the cells to differentiate. So after we lift the cells to the air contact, so there is at the basal level, we maintain the medium, and then we expose the cells to the air contact so that it develops as a lung interface, like air and liquid interface. So then we use this developed model. It actually needs to grow up to 21 to 30 days to develop to a completely differentiated air liquid interface model. So after that, we actually grow biofilms on the surface of them, and then that acts as a biofilm model, and then we use the antibiotics with the defined doses to see whether it actually works on the clinical pathogens or not. So some of the other facilities that we have to define some of the objectives are capacity to perform the genome-wide analysis, comparative analysis for the bacterial pathogens. And we have an in-house bioinformatic expertise. This is a co-facility between India and UK, that is a SCARAB and CMC Velo for rapid ID diagnosis and diagnostic metagenomics, and to identify competitive inhibitor. So for example, whenever we talk about antimicrobial resistance, so there comes the enzymes, so which actually help the pathogens to cleave off the antibiotics. So mostly it is enzyme mediated for gram-negative infections. So if there is a competitive inhibitor, so which can go and bind to the enzyme, so that we can prevent the cleavage activity, so that the antibiotic can go and do its job. So we are trying to identify a few competitive inhibitors for those resistant enzymes. And also we are studying network-based analysis for gene-to-gene interactions. So once we get the whole genome data, so what do we do is we see whether there is an interaction between the AMR genes and whether there is an interaction between the resistant genes and the susceptible genes. So that is how we link whether they are actually related or not. For example, I'll just give you a very short data. So what you see here is a phylogeny, core genome-based phylogeny of Klebsiella pneumoniae clinical pathogens. So what we can understand by doing this type of analysis, we can know, so whether this is a particular single species outbreak or it is a polyspecies outbreak. So for that, if we see the phylogeny, we can know how closely these organisms are related to each other, and whether the infections that are created in the patients are actually from the same source or different source. So based on the relation between the phylogeny, we can get to know that. And okay, so if we see the genes here, so the gene patterns also differ between the different clones. So clones I call group of organisms which club together, which are closely related. So if we see, we have two clones in this diagram here. So the top three organisms belong to ST1813 clone, and the bottom one belong to ST395. So this is a variant of 395. So what we see here is the genome configuration between these two pathogens for antimicrobial resistant genes. So we see that it actually matches together. So the organism found within a single clone will always like match together, so that we get to know that it is the same organism which is causing the infection. And we can also like look up the biofilm forming virulent genes, and we could also see that the biofilm forming virulent genes also match between the same clones. So this is one of the objective. So, and also recently we have been successful in winning a grant for the GCRF networking grant, so funded by GCRF and the Academy of Medical Sciences UK. So where we again formed an international consortium of biofilms and antimicrobial resistance of the ODA receiving countries, so official developer assistance receiving countries. So this actually a collaboration initiated by a collaboration between India and UK, that is the SCARAB and CMC facility. And as of now, we have 5 participants. So Thailand, Sri Lanka, Cambodia, Nepal, and of course, India. So due to the COVID impact, so we did not launch this network in full time. So, but we are looking forward to take it into full progress pretty soon. So we will definitely be expanding this network by including more and more experts from India and UK. I'm looking forward to see most of you there. Thank you.
Thank you. Thank you very much, Naveen. And I guess thank you to both yourself and Esther for describing so well the capabilities within the SCARAB facility. And I encourage people to reach out to you. We've got a couple of questions which we probably have time to get a quick answer to, and then before we move on.
Sure.
The first of these was, are you trying to look at true resistance versus tolerance to antibiotics in microbes within biofilms?
Yes, so we are actually looking at the true resistance. So we are not actually looking at tolerance only. So because I say this because like we actually check in vitro for the minimum inhibitory concentration of the antibiotic. So, and then we use the MIC levels into different levels. Like for example, say 2 times, 3 times or 4 times the MIC level to check what is the tolerance of the biofilms above to the MIC. So we check both hand in hand.
Great, thank you, Naveen. Actually, the second question is one for Esther. So Esther, perhaps you could type the answer to that in while we move on. So thank you very much, Naveen, for your presentation. And it's my pleasure to introduce our last speaker today, who's Dr. Sourav Ghosh, he's a lecturer in the Department of Healthcare Engineering at the University of Loughborough. And Sourav's going to talk to us today about something I know he's particularly passionate about, which is the rapid point-of-care phenotypic detection of bacteria and antimicrobial sensitivity. So Sourav, over to you.
Thanks, Mark. Is my screen visible?
Yes.
Mark?
Yes.
Thanks, Nihal. So yes, that's the topic I'm going to talk about. So good morning and good afternoon, everyone. I think I'll straight away move to my discussion. I must give a disclaimer at the beginning that unlike most of you here, I am not a biofilm specialist. I come more from an engineering background. I look at rapid diagnostic tests. I moved into, after a few years in the industry, I moved into this field of biomedical engineering. And I've been a member of faculty at Loughborough University for the last 7 years now. For those of you who are not so much aware of Loughborough as a place, it's more or less at the center of the country, and it's not too far from London. And Loughborough is how you pronounce it. I think that's where most of us make a mistake. Even in this country, people can't pronounce the word Loughborough. So this is quite a pretty campus where we work. And in terms of expertise, it is primarily engineering university. We do not have a medical science, a medical school here. And manufacturing, energy, and especially in sports science, it is doing quite well, I would say. And in our school, we have got EPSRC doctoral training centers, which are centers for training PhD students in the areas of regenerative medicine manufacturing, which is where I primarily started to work in biosensors. but we also have additive manufacturing and embedded intelligence. In our lab, we look at optical and acoustic sensing principles, and now we are also doing electrochemical principles. We also look at microfabrications, microfluidics, and try to understand more on the bio aspects of the problem by working with clinicians, with industry, people like Mark. We worked together when he was in Smith and Nephew on an antimicrobial resistance program. So our Center for Biological Engineering is quite unique for a university which is more focused in engineering. It has got 300 square meter of class 2 facilities. And I just mentioned about the EPSRC Center. And we have trained more than 100 PhD students in the last 10 years. We also led the EPSRC program on antimicrobial resistance, where I mentioned about Mark was on the board of governing body. And we have a doctoral training center in infectious disease, which I lead. I want to talk more about the relevant topic here, which is bacterial infection. And it's not so much of biofilms. I haven't done so much of biofilms, but hopefully with collaborations with the people like yourselves, we can move more into biofilms. But the paradigm change that we have been looking at is to go from that 72 timeframe window for a traditional diagnostics to a 1 hour window. The rapid tests, which are there, are primarily genotypic for bacterial detection, which are not definitive diagnosis, and definitive diagnosis through a phenotypic detection of bacteria and antibiotic sensitivity is what we were aiming at. The fundamental principle, I'll go very quickly here, is that a transducer, which is either electrochemical, optical, or electromechanical, in a single step binding, after a single step binding with the analyte, should produce the signal. That's what we aim at. This is a fundamental principle on which we work on. And there are various ways we can do that: electrochemical, optical, or electromechanical. I have been primarily working in the last two, and I will today I'll mostly focus on the optical, because this is where we have got most success in detection or rapid detection of bacteria and antibiotic sensitivity. This is the workflow, and this is an example for a primary care or even a secondary care urinary tract infection diagnostic test. It's slightly different in which the diagnostics, in which the primary care thing works in India and UK. But essentially, if a doctor were to straight away send the sample, this is essentially the workflow time. The actual time would be much longer because of the delays in transport. And what we are looking at is end-to-end within an hour, if we could go from detection to antibiotic susceptibility. And the basic principle in this particular case is an aptamer, which is an oligonucleotide, which has a fluorescence dye, which remains in a normal state, and it goes into, it stays in an off state in a normal state, and it goes into an on state after binding with bacteria. So after a single step binding, when the sample is introduced, it goes from an off to an on, so there is an increase in fluorescence. And then by introducing antibiotic into it, we can monitor whether the fluorescence keeps on growing, which suggests resistance to that antibiotic, or it decreases, suggesting that it is susceptible to the antibiotic. So that's the principle, and I'll show you some representative results. This is a kinetics, and what's happening is we, in synthetic urine sample, we introduced, we spiked various bacteria, E. coli, O111, and Staph epidermis and E. coli 055. So the red and the green one are the two serotypes of E. coli, and the blue one is the gram-positive version. This was an aptamer which was supposed to detect O111, but 055, not being too different from O111, also detects that. So this was the first proof of principle that within 5 minutes we are able to detect bacteria. And we did a serial dilution. We found that, how good are we without optimizing, and we could see that we could detect up to 1000 CFU per milliliter. So this is why we thought that we could probably target the UTI infection. And we also tested the antibiotic sensitivity, and we saw that when the antibiotic was introduced after 5 minutes, the signal went down, and within 1 hour, it was certainly visible. Even within half an hour, if we had measured, we could have seen the change. And without antibiotics, the fluorescence kept on increasing as the bacteria kept on increasing and bound with more and more aptamers, which went from turned off to a turned on state. The fluorescence increased. But one thing we noticed that when we tried with various serotypes of bacteria, the quantitative response was different. So O111, 055, Pseudomonas aeruginosa gave responses to different levels. O1 and O2 and O6 did not. Staph epidermis gave nearly no signal, or signal same to blank, which was encouraging. But all the other strains not giving signal was not very encouraging, and they were a mix of pathogenic and non-pathogenic. And then we figured out that it was because this aptamer, the receptor molecule that we chose, and we designed the aptamer beacon out of it, was targeting the whole of the lipopolysaccharide, which is the signature of a gram-negative bacteria. So what we did, instead of using that, we chose an aptamer which was targeting a conserved region of LPS instead of the entire LPS. And then we found much better results. We found that the quantitative response was more or less conserved across all gram-negative species. So this is one key result that we were very encouraged to observe, that we could differentiate between gram-negative and gram-positive, and quantitatively, the quantitative response was more or less similar across all gram-negative species. And we, through a project funded by NBIC, we also applied this to a wound mimic sample, working with Smith and Nephew, and we found similar response. Within 5 minutes, we could detect Pseudomonas aeruginosa in wound mimic sample. It was a buffer which mimicked the wound quite closely as recommended by Smith and Nephew. And once again, we could see that, yes, we could go up to 10 to the power 3, but this time the statistical significance was not so high. However, the specificity was quite good against gram-positive. Is it possible to do species-specific? In principle, yes. If we could design a beacon, probably not using an aptamer, we could either use an aptamer or we could use a peptide to make a beacon. And we have also tested peptide beacons. But here we presented primarily aptamer beacons, which were designed to detect any gram-negative bacterial species. We were more interested in this because the detection confirms the presence quantitatively of bacteria for any gram-negative bacteria. But this one we tried out with 2 different antimicrobial. So this is not, it's not antibiotic, this antimicrobial sensitivity. This was Acticoat and this was Iodosorb, 2 different antimicrobial agents supplied by Smith and Nephew. And again, we found, and this is the dose response as well, as Sanjeev was mentioning, is something similar to that. With different concentrations of Acticoat and Iodosorb, we observed when we see the response. We see that without Acticoat, the fluorescence kept on increasing, as happened without Iodosorb. And with different concentration of active code, we found when exactly the inhibition started. And we cross-checked and validated with plate culture results. So that was one part. And since it's only 15 minutes, I would very quickly say that in our group, we also, so we primarily work on developing new principles of detection. And this was one, which was based on a beacon concepts, fluorescence beacon. The other two are based on acoustics or electromechanical sensors. And the reason I want to present here is, again, these are some pioneering concepts, which is application of nonlinear acoustics for, again, one-step detection of microparticles, which works on the principle that when bacteria bind to the surface, it distorts the sinusoidal oscillation of a resonator. And in this case, it's an in-plane resonator. It's a bit of mechanical vibrations involved in this concept. And it is possible to mathematically separate the distortion by looking at the harmonics. And we do modeling and all that, which I won't talk about here. But we understand what we are doing. This is an example of a multidisciplinary project where we combined mechanical engineering with microfluidics, microfabrication, and of course, microbiology. And essentially, it is the magnitude of the third harmonic current which decreased. And again, I won't go into the depth of it, but it essentially meant that compared to the traditional acoustic methods, we could better differentiate between a specific and a non-specific binding, which is a challenge in direct detection. And we also found that by looking at this nonlinear the third harmonic current, which is the nonlinear response, we could more quantitatively detect bacteria, which was not possible in the traditional acoustic methods, where only shifts in frequency or shift in energy loss or dissipation are measured. And we also applied this to determine an acoustic signature of hematopoiesis, where red blood cells were grown from stem cells to mature blood cells. So we applied this in various biological scenarios, from cells to bacteria. And we look at developing integrated sensors using this acoustics, where we go from collection. And this is an example from an EU project that we did, which collected the breath and then captured the virus on microfluidic devices, which had the acoustic sensor. And then we actuated and sensed. So it was more like an integrated biosensor example. And this is how we do it in the lab. We integrated everything, the bacteria, the microfluidic chamber, the fabricated device, and this nonlinear network analyzer that we have built. We are quite proud of this. This is a very unique network analyzer, which determines or which measures at 3 harmonics simultaneously. And we have sent a few of these across the world to Stanford, KU Leuven, Leicester. And
Sure, I'm going to have to ask you to wrap up in a moment, if that's okay.
Yes, yeah. I'm just saying that, and this was another one in acoustic biosensors. And the reason I present this is I heard about quorum sensing. So this is another method that we have applied for quorum sensing, which I haven't presented here, but I'm very happy to share that. And thanks for listening, and very happy to discuss about how we can work together.
Thank you very much, Surab, for a fascinating talk, and thank you very much for all our speakers today for their excellent presentations, and for all our participants and attendees. Surab, there is a question for you in 1 or 2 in the chat, which I would ask if could you answer those, type those in, perhaps?
Yeah,
while we're
sure. Yeah.
I just wanted to say thank you to everyone for your participation today. Thanks to our speakers, to the attendees, and to our colleagues in the India Biofilm Society for allowing us to engage with you.